So, plasma membrane. Yep. Okay, these are the outer membrane of all cells. Okay, doesn't matter whether it's prokaryotic, eukaryotic, plant, animal, fungus, protist, archaea, bacteria, they got a plasma membrane, okay? And the plasma membrane, okay, we're going to talk about its structure and kind of its function, all right? But the function, first of all, again, some of this we're already rehashing, but it is selectively permeable. Can anybody tell me what selective permeability means? Basically, it just means that it lets some things in, but not all things, okay? It picks certain things that can come in. To permeate means to go through, okay? So to be selectively permeable, it allows only certain materials through. Okay? So that's the main thing of the plasma membrane. It keeps things out, lets certain things in, keeps certain things in. Alright? So let's talk about the structure of the plasma membrane. Okay? Okay, so the structure of this. The structure, the most basic definition of this is that it is a phospholipid bilayer. Okay? So that means it's bilayer. Bi means two. Two layers of phospholipids. Okay? Phospholipids are generally drawn, again, like that. And so if we were going to draw a quick little example of a phospholipid bilayer, it looks something like that. Okay? Two layers with these. Okay? Now what makes up lipids? We've got the head. What is the head made up of? Does anybody remember? Should be in your macromolecule notes.
Is it the fatty part?
No, that's the tails.
Oh.
This is the G part. Glycerol. Okay, so it's got a glycerol head. Okay, and like Dylan said, it's got the fatty acid. You guys probably can't see it underneath that. Tail. Okay, so with this, the head and the tail both have specific things about them. All right, so the glycerol head is hydro. What does hydro, the prefix, mean? Water. Okay. Hydrophilic. Okay. So Philadelphia, what do they call Philadelphia? The city of brotherly love. Okay. Phila is Greek for love. So hydrophilic. They love water. Okay? Hydrophilic. This just means that the glycerol head attracts water. Okay? So water will come in to the phospholipid bilayer. Okay? So you can look at it. We've got water. Okay? That water is going to come in through the heads. Alright, so once it's in here, now it's near the tails. Alright? The tails have their own property. Okay? Fatty acid tails. All right? These are hydro. Yep. Which means they hate water. Phobias. You're scared of something, okay? You don't like it. So these will repel the water. Okay, but since the water's already coming through, okay, if these were hydrophilic, these tails, it would just kind of sit in between here, right? But what do you think it does to the water?
It pushes it either direction.
Yeah, it can push it either direction, but since it's already come through here, and these ones want the water, they like water, right? The water's just going to go whoosh really fast, okay? And come through the phospholipid bilayer, all right? Water can do that, other molecules can't, okay? So if it's bigger molecules, like salt molecules, certain proteins, those don't make it through the phospholipid bilayer.
Wait, so then the glycerol head has to be like way stronger than the fatty acid tails. Like, doesn't it like pull it all the way through?
Kinda, yeah. But so it's pulling down here, but this is also like pushing, okay?
Oh, I see.
This is pulling and this is pushing.
But then those are also pushing away at the same time. The ones on the bottom. The ones that are pointing up, aren't they pushing away?
Yeah, you're correct. The heads are a little bit stronger than the tails in terms of traction versus the repulsion in the middle. But just know, the reason that water doesn't get stuck in the middle basically is just because these things don't want water there, okay? The tails don't want water. The heads like water. So, once it passes through one head, it's making its way fast to the other head because these don't want it there. Does that make?
Yeah.
Okay. So, this is the most basic structure we've got, okay? But there's a few other things in our plasma membrane, okay? One thing that is inside of this cholesterol, okay? Cholesterol sits inside of our, cholesterol sits kind of like, I'll use a red one, okay? I'll just draw like a box, okay? Oh, it's bigger than that, cholesterol's bigger than that. Okay, those are cholesterols. Okay, so this cholesterol, what its job is to make sure that the tails do not stick together. Okay? Cholesterol keeps fatty acid tails from sticking together. Okay. Is there any questions about any of that so far? So the meat and potatoes of it is this bilayer, the phospholipid bilayer. But then you have some of these cholesterol scattered about to make sure that there isn't sticking. And there is one more thing to find in there. Wish I had a purple marker. We used to have purple markers. Purple markers were great. They erase so well. Alright. And so the final thing that we're going to talk about are basically these big protein shoots, okay? And these are called transport proteins. Okay, so you got transport proteins. Anybody got a guess as to what these proteins do?
Transport something.
Yes. So water can make it through the bilayer, it just kind of moves back and forth, right? We understand that, that water has the capability. But what if you want to get things in or out of the cell that are not water?
You just have yourself a transport program.
You have to use the transport proteins, okay? So this is essentially the, I don't know, think of it like the Panama Canal, right? That's how boats get across between the two oceans right there. There's a canal that kind of pumps them through. Same thing, transport proteins move molecules that are too large to cross the bilayer. Okay? Does anybody have any questions about that? Alright. So let's go ahead. Okay. So now we're going to talk about, we're going to start what we will be doing our lab about here. Okay? So,
Do you want this in lab notebooks or?
Nope, we're not putting your lab together yet. We're just talking, this is normal notes, and then I'll put the stuff for your lab notebooks up there, okay? After we get this done. So passive, cellular, transport. Okay? So what do you think passive transport uses instead of active transport, or doesn't use?
We talked about this last year, didn't we?
Probably not.
I don't remember talking about this.
I can't imagine we would have talked about this last year in earth science. But anyway, passive versus active. Which one of those do you think uses energy? Active, okay? So passive cellular transport doesn't use energy. Okay, this goes with the flow. Probably the last time you would have talked about this would have been Keegan. Maybe we would have talked about it in 8th grade in physical science a little bit.
I don't remember talking about this.
But either way. So passive cellular transport doesn't use energy, goes with the flow, okay? What cells are trying to get to when they do this is called dynamic equilibrium. Okay? So equilibrium is kind of when all things are balanced, right? And what you're trying to do here with dynamic equilibrium is reach a point where concentrations of molecules is equal inside and outside of cell. Okay? So you're trying to get it where everything's kind of balanced. Has anybody ever had to get an IV before? What are they putting into you to keep you hydrated when they have an IV in you? Does anybody know? It is not water, and we'll see why it is not water here in a second. It's a saline solution, okay? There's some salt in that. And that's because our cells have about, what is it? I think it's 0.8% salt inside of them, like by concentration, okay? And so if you were to put something that was 100% water in there, okay, that cell is going to try and get it equal, okay? It's going to try and take in a bunch of water to lower the salt concentration inside of it, okay? What happens if your cell takes in too much water, do you think? Okay? Yeah, you'll have a bunch of cells popping in your body. Okay? On the flip side, if there's, say you were to use like a 10% salt solution for your blood, if you're trying to hydrate yourself with that.
Is it a lot or too much?
Well, considering 0.08 is what they use, or 0.8 is what they usually use, it's a lot. So, if you're using a 10%, the cell's going to try and equal it out and try and push a bunch of water out of the cell to try and equal it what's outside, right? So this is kind of, and again, this is what the lab we're going to do. You'll see this, okay? By pushing water through, it basically shrivels up, okay? So you don't want your cell to shrivel up, because then it doesn't work very well. And you don't want your cell to get too full of water, because then it'll explode, which then it doesn't work very well either.
So like, when you chug water, like your cells like full up or what?
No, because your body has a process. You have natural salts in your body from the things that you do. That's like what happens when it filters through your bladder.
Well,
and your
but
kidneys
it's
and all
when
that.
like it's going straight into you.
Yeah, that's when you're talking about an IV. Intravenous is what IV stands for, so it's going right in your veins. There's no process where it's going. So they have to give you that saline solution, okay, to make sure that your blood is the right amount of salinity, the right amount of salt. Okay? So we're always trying to reach this dynamic equilibrium. Okay? And the ways that we do this through passive transport, there's a couple of them. Okay? But we're going to talk about the overall thing is diffusion. Okay? And so diffusion is very simple. Okay? All you're doing with diffusion is you're moving from areas of high concentration to low concentration. Okay? So, like, here. Again, I'm sure you guys all understand this, but visuals never kill anybody. Okay, so this is just 100% water right now, right? Well, that's probably not true. There's probably some minerals in there. We're going to put this food coloring in, okay? Now, right now, this food coloring is not equally distributed, right? It's kind of, there's some splotches where there's more, some splotches where there's less. But after we give it a few minutes, or if we stir it around, it's going to go to the areas where there's really high concentrations of food coloring, right? It's going to distribute out, it's going to diffuse throughout to the points where there's lower. And now it looks relatively homogeneous, right? It looks pretty much the same color. If you were to add more food coloring even to this, there's areas in there that look thicker still, right? That has to diffuse out. Okay, if you kept adding more and more food coloring, this would get darker and darker. Okay? Can also think of it, so like the 7th graders from last year, the 8th graders, notorious farters. Disgusting. They were gross. And they all thought it was so funny. Like they bragged. Now say we sat Kellen back where Jack is. Kellen was one of the worst.
No thanks.
Kellen lets one rip back there, gases also diffuse out. And trust me, there was a lot of gas. So eventually it would make its way over the whole class because there's area where there's more fart, the high concentration areas, and less fart, until there was equal fart everywhere. Thank God I had them last period of the day so nobody else had to come in for that after them. What a joyous, joyous class that was. Okay, so does everybody get diffusion? We good there? Okay, now we're going to talk about two different types of diffusion. Okay? I think that's all that's in this passive transport. Yep. Yeah, perfect. Okay. So with inside of diffusion, the one that we're going to be doing, and the name of this lab will be called the Egg Osmosis Lab. So we need to talk about.
Is this our?
No, this isn't a lab yet. This is still notes. Okay. So osmosis. Osmosis is very similar to this, but it's just the diffusion of water across a selectively permeable membrane. Okay? So this is just the diffusion of water, not the diffusion of, food coloring or gas or anything like that. We're specifically talking about water, and we're specifically talking about how it moves across membranes. Okay? It's not always going to be with like the cell membrane or with the egg membrane. Sometimes it's talking about like a filter paper. Okay? But water moves to try and reach that dynamic equilibrium. Okay? It wants to get it equal everywhere throughout. Alright? And again, I could kind of go into more depth about this, but it's going to be more evident when we just start doing the lab and you can kind of see what's happening. Again, this is going to be a process where it's going to be more than one day. Generally, this lab takes about 3 days, but I think it'll probably only take 2 with the way that I've got it set up.
3 days because you have to wait, or 3 days because
You're waiting. Well, you have to wait because it takes a while.
Well, yeah, I know, but like 3 days, you get through all this stuff.
What? No, and it's not because there's a lot of stuff. Like each day only takes about 15 minutes, but you have to wait like a 24-hour period to get the right amount of things. I don't know. Who knows? On Wednesday, you guys should come in here and do all your stuff with the eggs and put them in water, because maybe by the end of 3 hours, they'll do what they're supposed to do. So, we'll see. I'll leave that. Dylan, stop messing with it. This is why they fall. Alright, so osmosis, we'll talk more about that. And then the other one that we have is facilitated diffusion. Okay? And so facilitated diffusion, this happens only in cells, alright? But this is where you use transport proteins to move molecules through membrane to reach dynamic equilibrium. Okay, so generally it's water that's moving across the membrane, but sometimes we try and pump out sodium, or we try and pump in potassium, okay? We're trying to make it so that it's equal, all right? Generally water moves, because water moves a lot faster, but sometimes we try and facilitate the diffusion by pumping out larger molecules to try and get to that dynamic equilibrium. Okay? Hypotonic solution. Okay, so a cell is said to have a hypotonic solution inside of it. And just really fast, do you guys remember what like solutes and solvents are? I should probably just rehash that real fast. Solute is the dissolved, one S in dissolved, material in a solution. Okay, so an example of a solute would be like salt and a solvent would be the material that dissolves solute. Okay, so that would be something like water. Okay. So, a hypotonic solution. This is where there is a higher concentration of solute inside cell than outside. Okay? So if there's a higher concentration inside than outside, what's going to happen to that cell if it wants to get equal? So there's more in here. But we can't pump out, let's just say salt. Okay? We can't pump the salt out of here to get it equal. So what are we going to try and do to get those concentrations equal? If we can't put salt out, what are we going to put into the cell? More water to lower that concentration. Okay? So, water moves into cell. Cell swells. And sometimes bursts. Okay? Does everybody have the solute and solvent stuff down, or can I erase that? Okay. So I want to put some visuals on the board. Bless you. Alright, so in the middle here, we're going to put our normal cell. Okay? Just a normal cell. Over here, this is our hypotonic. Okay? One of the ways, and I don't know if this is going to help you at all, but again, this is just something that kind of helped me. It's stupid, but the way I remember the difference between hypotonic and the other one, hypo, okay, because the other one's called hypertonic, but hypo makes your cell look like an O, okay? Don't know why I remember it that way, but I do. Okay, hypo, basically just whoop, cell swells. Okay? Now a hypertonic, it's not 2 words. I don't know why I keep writing it's 2 words. Okay? What do you think that'll end up looking like?
Shrivel up.
Yeah, it's going to be all shriveled up little thing. Okay? And so if there's higher concentrations inside than outside in hypotonic, what do we have in hyper? Exactly. Okay? There's a higher concentration outside of cell than inside. Okay? So we have a higher concentration outside than inside. So water, instead of water moving in, water moves out. Cell shrivels. Okay? So we have hypotonic, hypertonic, and now we need to talk about this one, the middle one, the normal one, where you have equal concentration on either side. Okay? Okay, isotonic. Okay. So in an isotonic solution, if a cell is in an isotonic solution, this means there is equal solute concentrations inside and outside of cell. Okay? So this would be what would happen if you used 100% water when you were giving someone an IV. Over there, hypertonic would be if you gave somebody like a 10% saline solution. This is our happy little medium. Okay? This is why we don't use straight water and why we don't use a bunch of salt. Just a little bit of salt to make your cells happy. Keep them in that dynamic equilibrium.
So hypertonic cell is just basically a dehydrated cell.
Yep, and this is an overhydrated cell. So
you got raisins and grapes.
well, like super grapes. Active transport in cells. Oh, we should probably do that too. Yeah, we're gonna have something after this. I'm just gonna go over all the functions of the organelles to make sure that everyone's on the same page. Even though I'm sure you guys probably all got the right information, I just want to go over it real fast to make sure that we're all good. Okay, so active transport in cells, there's three main ones, okay? So there is endocytosis. Okay? So when we talk about endocytosis, this is also referred to as cell eating. Okay? Basically what happens in endocytosis is a large molecule pushes its way into the cell, taking part of the cell membrane with it. Okay, so on like a three-step thing, it would kind of look, we've got our cell, okay, and you got your little other stuff. All right, so it's going to be moving. Part one, then part two. It starts to push its way into the cell. Okay, it's pushing, pushing, pushing. Eventually, it kind of just goes and pops right off. And so then you're going to have step three. The thing essentially just kind of made its way in, took part of the cell membrane, okay, because this will close around it, okay, it'll close around it like that, and then it's inside of the cell, okay. The cell kind of looks like it's eating it. Another way that I remember this, so things are going into it, into endo, kind of sound the same. I don't know. If it helps, it helps. If it doesn't, it doesn't. All right, any questions about endocytosis? Exocytosis is pretty much if you did this in reverse, okay? Yep, it's exiting. Okay, so exocytosis, large molecules, leave cell, okay? And usually these are in these things called vesicles. Okay, vesicles are made of same material as cell membrane. Okay, and so basically they just kind of vesicle attaches. There's not an extra T in there. To cell membrane, releasing contents. Okay. And so, if you're going to draw a picture of this, again, pretty much the opposite of that. You've got whatever's inside of your vesicle, okay? Step 2, the vesicle begins to become part of the cell membrane on one side, and then it opens up, releasing that thing, okay? And then we'll just eventually go to a normal cell with whatever's released, okay? So it's basically this but backwards, okay? Anybody got any questions about either of those? Alright. Give you guys a second to get that all written down, and then we'll talk about the final thing in terms of all this stuff. Sodium potassium pump. A lot of fun. Again, this is a little bit more complicated than the other ones we talked about, but it's not terribly, terribly complicated. This is a pretty good video that should help us kind of understand what's going on a little bit better. Better visual than anything I can draw. Okay, so the last one that we're going to talk about today is the sodium, potassium pump. Okay, now for this one, we are going to be using a regulated transport protein. Okay, and all that it means when it's regulated is that it means something has to attach to open and close it. energy that this uses is called ATP. This uses ATP. ATP stands for adenosine triphosphate. Okay? And that's basically the main thing of energy. If you remember ATP, that's going to be one of the most important things you learn about during this class. Alright, so basically what this is doing is it uses energy to bring sodium into cell and get potassium out. Okay? Because we have high concentrations of these two things. Okay? Alright. So the first thing that is going to happen that's a little sodium potassium pump. Come on. It's got sodium monoxide, potassium monoxide 0. Oh, I said that backwards. Ugh. It's not bringing sodium in, it's to push sodium out. Because you make a lot of sodium and you need potassium. Excuse me. So, hopefully you're not writing in pen. If you are, just scratch that out. To bring potassium into the cell and get sodium out. Ugh. Sorry, it's been over a year since I taught this. Okay, so anyway, your first step is sodium from inside cell binds to transport protein. Okay, so that's the first thing you're going to do. You're going to get, and this is 3 sodiums. Okay, for every 3 sodium you push out, you take 2 potassium in. Alright, so that was pretty quick, but second thing that happens, ATP binds to transport protein. Okay, so when it binds to that transport protein, it's going to start the process of opening this up. Alright? ATP breaks off and leaves one phosphate attached to protein. Okay? And so, the real energy, where it stores energy in ATP, is in that bond between the second and third phosphates attached to it. Alright? When that bond gets broken, it releases a lot of energy. And you're going to create a single phosphate and then what's called ADP, adenosine diphosphate, where there's only 2 phosphates, okay? So once we release that, okay, that energy that gets released will allow the protein to open, okay? So, step 4, energy released from ATP breakdown, opens protein to outside of cell, and releases the 3 sodium. Okay? So right here, it opens out and the sodium leave. Okay? Now this is open and we want to bring potassium in. Okay? So, step 5. Potassium, sorry, 2 potassium, bind to open transport protein. Alright, so now we just have to get these inside the cell. Alright? So what we're going to need is to release that phosphate. Okay? Step 6. Phosphate releases from protein opens to inside of cell, potassium is released into cell. Okay, it's kind of complicated, but basically sodium wants to get out of the cell, potassium wants to get into the cell, so we attach it, we break apart an ATP. Once that ATP breaks apart, enough energy, it opens and releases the sodium. When that's open to the outside, potassium binds, the phosphate leaves, which basically like switches it so that it opens back into the cell and the potassium then comes out. Okay. Are there any questions about that? On your guys' quiz, there'll probably be like 1 question about the sodium potassium pump. Okay? Something to know that that happens, but it's not like crucial. Okay? The osmosis diffusion stuff is more important. Alright? Are there any questions about any of this? Sorry, you just comment like that, so I wasn't sure if you had a question. Okay. We've got about 45 minutes. We're going to go over the organelles and stuff, and then I'm going to have you guys wipe down your desks. But let's go ahead. It's pretty nice outside right now, so let's go spend about 10 minutes outside. We've been going pretty heavy on the lecture stuff, so make sure you got your masks on.